Ever wondered how plants turn invisible carbon dioxide into sugars that power life? Hi, I am Dr. Mashita Pise, and in this episode of Biochemistry Insights, we will uncover Kelvin cycle, the dark phase of photosynthesis, where ATP, NADPH, and a little magic of enzymes transform air into energy. So let's see. Dark phase of photosynthesis occurs in the stroma of chloroplast. Now you can see here that leaves contain mesophyll cells, and inside the mesophyll cells we have chloroplast, and the fluid portion of the chloroplast is known as stroma, where the dark phase of photosynthesis occurs. Now dark phase reactions, they are independent of light, but they require ATP and NADPH produced during the light reactions. The main function is to convert atmospheric CO2 into carbohydrates, and it was discovered by Melvin Kelvin in the year 1961, who received Nobel Prize for this particular discovery. This pathway is also called C3 pathway because the first stable compound in this cycle is a 3-carbon compound known as 3-phosphoglycerate. Let us now understand the dark phase reactions in detail. Don't be misled by the name. It's not about happening only in darkness. These reactions can occur during the daytime as well as during the nighttime. Instead, it uses the products of light reaction, that is ATP and NADPH, for fixing CO2 into organic molecules. Dark phase reactions have 3 important stages. The first stage is known as carboxylation, wherein CO2 is fixed, and 3-phosphoglyceric acid, that is 3-PGA, is produced. In the second step, which is a reduction step, 3-PGA is converted into glyceraldehyde 3-phosphate using ATP and NADPH produced during the light reactions. The third and final stage, regeneration, involves regeneration of RuBP so that it can be again utilized for the carboxylation stage. Now let us understand energy budget of the Kelvin cycle. For each glucose formed, 6 CO2 molecules, 18 ATP and 12 NADPH are required. The products of dark reaction include glyceraldehyde 3-phosphate. This is used for glucose, sucrose and starch production, and along with this glyceraldehyde 3-phosphate, ADP and NADP is also produced. Without ATP and NADPH of the light reactions, the dark phase reactions would stop completely. The overall reaction of Kelvin cycle is 6 CO2 plus 18 ATP plus 12 NADH will be converted to glucose molecule plus 18 ADP, 18 inorganic phosphate and 12 NADP. Let us now understand the 3 stages of dark reaction in detail. As I said, dark reactions, they have 3 stages. The first stage is carboxylation, wherein the enzyme RuBP carboxylase and oxygenase, which is designated as RuBisCO, fixes CO2 into ribulose 1,5-bisphosphate, and this results in formation of 3-phosphoglyceric acid. Remember, 3-phosphoglyceric acid is a 3-carbon compound, and this is the first stable compound of this cycle. Therefore, this pathway is also known as C3 pathway. Now let us understand this whole step. Ribulose 1,5-bisphosphate, RuBP, can tautomerize between keto and enol form. It is in the enol form the enzyme adds CO2 to RuBP, converting into a intermediate which is a beta keto intermediate of RuBP. Now this beta keto intermediate of RuBP undergoes hydrolysis yielding two molecules of 3-phosphoglycerate या फिर 3-phosphoglyceric acid which is PGA, 3-PGA. So you can see here it's a 6-carbon molecule. It splits into 3 carbon molecules and 2 copies of it, 3-PGA. Okay, so 3 molecules of RuBP rearranges to form the enol form of RuBP, wherein 3 carbon dioxide is fixed, converting into 6 molecules of 3-PGA. In the second step, the output of the carboxylation reaction, that is 3-phosphoglycerate or 3-PGA, is converted to glyceraldehyde 3-phosphate. So 3-PGA or 3-phosphoglycerate is converted into 1,3-bisphosphoglycerate BPG first by the enzyme phosphoglycerate kinase. So this is a common reaction from glycolysis or any other pathway. So you will be easily remembering this enzyme phosphoglycerate kinase. So what happens to PGA? PGA becomes 1,3-bisphosphoglycerate. Here ATP is hydrolyzed. So this is the step where ATP of light reaction is hydrolyzed. Okay. Now let us see what happens to 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate is then reduced to glyceraldehyde 3-phosphate, that is glyceraldehyde 3-phosphate, or usually commonly it is written as GAP or GAP. Okay. So what has happened to PGA? 3-phosphoglycerate is first converted into 1,3-bisphosphoglycerate. ATP hydrolysis occurs, therefore additional phosphate group is attached to this molecule. Now the 1,3-bisphosphoglycerate gets converted into GAP, that is glyceraldehyde 3-phosphate. Here the second product NADPH of the light reaction is also utilized for the reduction. So what happens during reduction? Conversion of 3-phosphoglycerate into glyceraldehyde 3-phosphate GAP. Okay. So now you must understand that for fixing CO2 and producing one molecule of glucose, carbon dioxide fixation occurs 3 times. That means 3 molecules of RuBP fixes 3 molecules of CO2 and we get 6 molecules of PGA. Okay. So when PGA is reduced to glyceraldehyde 3-phosphate, we have 6 molecules of GAP molecule. Okay. So now after this step, each glyceraldehyde phosphate 3-phosphate molecule has a different fate. हमारे पास 6 glyceraldehyde 3-phosphate है और 6 में से हर एक glyceraldehyde 3-phosphate का जो फेट है इसके आगे के जो conversions है वो अलग है. To repeat in English, we have 6 molecules of GAP molecule, glyceraldehyde 3-phosphate, and each of these 6 glyceraldehyde phosphate undergoes different types of reaction after this step. Okay. So let us take the first 2 glyceraldehyde phosphate, which is converted to dihydroxyacetone phosphate, DHAP. What is the enzyme? Triose phosphate isomerase. Once again, a very common enzyme from the glycolysis pathway. Next, DHAP and 1 mole molecule of glyceraldehyde 3-phosphate react together. The enzyme is aldolase and we get fructose 1,6-bisphosphate, a 6-carbon molecule once again. Now this fructose 1,6-bisphosphate is then converted into fructose 6-phosphate by the enzyme fructose 1,6-bisphosphate phosphatase. It is a dephosphorylation reaction which results in fructose 6-phosphate. So what happens during step 7? Aldolase consigns GAP and DHAP into FBP, fructose 1,6-bisphosphate. Now fructose 1,6-bisphosphate is dephosphorylated into fructose 6-phosphate. Shall we move ahead? In step number 9, you need to understand that we have fructose 6-phosphate from the previous reaction. Now fructose 6-phosphate has 6 carbon. Okay. Now what happens? From the previous 6 glyceraldehyde 3-phosphate molecules, 6 gaps, okay, 2 we converted into DHAP. Now we are utilizing one more gap molecule, and part of the fructose 6-phosphate is transferred to glyceraldehyde 3-phosphate, creating, we have erythrose 4-phosphate and xylulose 5-phosphate. Okay, so see here, fructose 6-phosphate 6 carbon plus glyceraldehyde 3-phosphate we are using here, so 3 carbon. So 6 plus 3, 9 carbon. Okay, so this 9 carbon molecule splits into erythrose 4-phosphate and xylulose 5-phosphate. Okay, this is the reaction. Now, out of the 2 DHAP formed, 1 DHAP along with erythrose 4-phosphate produced during the previous reaction, again the enzyme is aldolase, we find pseudoheptulose 1,7-bisphosphate SBP. Okay, so remember, in the initial time we produced 2 DHAP, 1 DHAP merges with erythrose 4-phosphate, reaction catalyzed by aldolase enzyme, and we are forming pseudoheptulose 1,7-bisphosphate SBP. As soon as SBP is formed, it is dephosphorylated by a phosphatase into pseudoheptulose 7-phosphate S7P. So, how do we remember this reaction? Aldolase combines erythrose 4-phosphate and 1 DHAP into pseudoheptulose 1,7-bisphosphate. This SBP then converted into pseudoheptulose 7-phosphate. Let us move ahead. Now pseudoheptulose 7-phosphate S7P is then converted into ribose 5-phosphate and xylulose 5-phosphate. So how this occurs? One pseudoheptulose combines with one more molecule of gap, that is glyceraldehyde 3-phosphate, and again we have a 7 carbon compound, and then we have a 3 carbon glyceraldehyde 3-phosphate, total 10 carbon. Okay, so this molecule gets splitted into ribose 5-phosphate and xylulose 5-phosphate. So it's a group transfer which creates 2 new molecules as shown here. So now we have ribose 5-phosphate, xylulose 5-phosphate. Now ribose 5-phosphate immediately isomerizes into ribulose 5-phosphate. In this step, phosphopentose isomerase converts R5P into RU5P. So what we have now? Both the molecules are now RU5P, ribulose 5P. So you can see here, ribose got converted into RU5P. Now the second molecule, xylulose 5P, is also getting converted into RU5P. Okay, so phosphopentose isomerase converts 2 molecules of XU5P, xylulose 5-phosphate, into 2 molecules of ribulose 5-phosphate. Now finally, ribulose 5-phosphate is then phosphorylated to form RuBP, ribulose 1,5-bisphosphate. Remember this compound? This is our the starting compound which is used for fixing CO2, RuBP, by the enzyme RuBisCO. So once again, let us understand. Phosphoribulokinases, the enzyme, uses 3 ATP to phosphorylate 3 Ru5P into 3 RuBP. This is the regeneration step of ribulose bisphosphate. These are same 3 RuBP molecule that we started with in step 1. So the Calvin cycle is now complete. Are you bothered by such an elaborate reactions? Don't worry. I have made it simplified. Let us do it one more time. Okay. So remember, we started with 3 molecules of RuBP. RuBP oscillates between keto and enol form. It is the enol form to which 3 molecules of carbon dioxide are fixed by the enzyme RuBisCO, RuBP, carboxylase, oxygenase. Okay. As soon as 3 molecules of carbon dioxides are fixed, it forms an unstable intermediate which immediately hydrolyzes into a stable 3-carbon compound called 3-phosphoglyceric acid or 3-PGA. How many? 6 PGAs. Okay, 3 PGAs. Now, here the first carboxylation stage is complete. We have converted RuBP into 3-phosphoglycerate or PGA. Now, this is the second stage, that is reduction. What is reduced? PGA. Okay, how? This is reduction stage. We are using the products of light reaction, ATP and NADPH. Let us understand the second stage. 6-PGA is first phosphorylated using 6 ATP from the light reaction, which results in the formation of 1,3-bisphosphoglycerate or 1,3-BGP by the enzyme phosphoglycerate kinase, an enzyme from glycolysis pathway. This enzyme works there also. Okay. Now, what happens to this particular 1,3-BGP? This 1,3-bisphosphoglycerate is then reduced and also converted to 6 molecules of glyceraldehyde 3-phosphate or GAP. In this reaction, 6 NADPH is reduced to 6 NADP by glyceraldehyde 3-phosphate dehydrogenase enzyme. So here we complete the second stage as well. Okay. So RuBP plus CO2 gives you 6 molecules of PGA. Carboxylation over. Second stage, 6 PGA first converted into 1,3-bisphosphoglycerate and then formation of 6 GAP molecule. Reduction stage over. Now from here you need to understand what happens to these 6 molecules of glyceraldehyde 3-phosphate. So just now we completed the second stage, that is reduction of 3-phosphoglycerate into glyceraldehyde 3-phosphate. So we have now 6 molecules of glyceraldehyde 3-phosphate. So after this, these 6 glyceraldehyde 3-phosphate molecules each have a different fate. So let us understand this. As soon as glyceraldehyde 3-phosphate is formed from the reduction stage, one molecule of GAP goes out of the cycle towards formation of sucrose. It is this GAP which is now will be converted into carbohydrate. Okay. Rest of the 5 actually is utilized for continuation of the Calvin cycle. Let us see what happens to the rest of the 5 GAP molecules. Out of the 5, 2 more is converted into 2-dihydroxyacetone phosphate, 2 molecules of DHAP. Out of 2 DHAP, 1 is immediately converted along with 1 more molecule of GAP into fructose 1,6-bisphosphate. So 3 carbon from DHAP, 3 carbon from one more gap glyceraldehyde 3-phosphate, and we create a 6 carbon fructose 1,6 bisphosphate molecule, which is then converted into fructose 6-phosphate. Here, as soon as fructose 6-phosphate is formed, we use the 4th gap molecule to form 2 compounds. So remember here, fructose gives you 6 carbon, glyceraldehyde 3-phosphate gives you 3 carbon. So total we have 9 carbon in hand, and it is converted into 2 compounds. One, erythrose 4-phosphate, a 4 carbon compound, and xylulose 5-phosphate, a 5 carbon compound. Okay. We are interested in this, erythrose 4-phosphate. What happens to this? Out of the 2 DHAP, 1 is left. That 1 DHAP combines with erythrose 4-phosphate and forms a 7 carbon compound, pseudoheptulose 1,7 bisphosphate SBP, which is immediately dephosphorylated to form pseudoheptulose 7-phosphate S7P, pseudoheptulose 7-phosphate. This S7P combines with the last molecule of gap, glyceraldehyde 3-phosphate, forming 2 5 carbon compounds. 7 comes from this compound, 3 carbon comes from gap. So we have 10 in hand. So it splits into 2 5 carbon compound, ribose 5-phosphate, xylulose 5-phosphate. Ribose 5-phosphate is immediately converted into 3 molecules of ribulose 5-phosphate. Okay. And this xylulose 5-phosphate is also converted into ribulose 5-phosphate. Now both these compounds result in ribulose 5-phosphate, which is then phosphorylated using 3 ATPs into ribulose 1,5 bisphosphate or RuBP. So this RuBP is now regenerated, ready to undergo carboxylation one more time. So that's how dark cycle or dark phase Kelvin cycle occurs. 3 stages: fixation of CO2 first, then the product of the carboxylation reaction 3-PGA is then reduced to glyceraldehyde 3-phosphate, that is GAP. Then GAP is converted into sucrose, 1 molecule. Rest of the molecules are used in regeneration of RuBP. So this is how it looks. CO2 is fixed by RuBisCO enzyme into RuBP stage 1, forming 3-PGA. Then this 3-PGA is first phosphorylated using ATP and then reduced using NADPH into glyceraldehyde 3-phosphate. Part of the glyceraldehyde 3-phosphate goes into sucrose formation. Rest of the molecule is utilized to regenerate RuBP back so that this dark phase can continuously occur. So, what is the significance of dark phase reactions? Kelvin cycle is the primary carbon fixation pathway in most plants. It provides organic matter for the plant growth and food chains. Some of the key enzymes of this cycle are activated by light. Therefore, light has an indirect control. The main enzyme RuBisCO, its activity is influenced by CO2 and oxygen ratio. Remember, it is RuBP carboxylase oxygenase enzyme. That means if carbon dioxide is higher, then the carboxylase activity will be favored, and vice versa. It can happen during day and night both. If ATP, NADPH are available in ample quantity, it can happen even in the during the daytime. And that's the incredible journey of Kelvin cycle, turning carbon dioxide into sugars that fuel our planet. I'm Dr. Mashita PC and you have been watching Biochemistry Insights. If you found this helpful, Do not forget to like, subscribe and share. I will see you in the next lecture. Thank you.